Hello everyone, this is Supreet Kadakol, Assistant Professor, Department of Zoology, Sri Venkatramana Swami College, Bantwal. In this session, mainly we'll discuss about the final year paper 6 syllabus, that is genetics, biostatistics, evolution and paleontology. So same as paper 5, in this paper also we'll be having 4 units and under that many subunits. So we'll see the unit 1. In unit 1, genetics, the first subunit, it consists of introduction and laws of inheritance. So what are the branches of genetics, what is genetics, and little about heredity and variations, Mendel's experiments like monohybrid cross, dihybrid cross, test cross, back cross, and Mendel's law of inheritance. Along with that, use of Drosophila in genetic studies, why Drosophila is important for the genetics, and dihybrid crosses in Drosophila. So this will be completed in your first subunit. Whereas in second subunit, interaction of genes, very important subunit, where you will be studying about different types of interactions like in complement dominance, then plumage pigmentation in fowl, supplementary factors, that example is comb pattern in fowl, dominant epistasis, plumage color in leghorn and vandicott, coat color in dogs. These are the examples. And recessive epistasis, where we'll be studying about coat color of guinea pigs. Complementary factor, the example will be flower color of sweet peas. And lethal genes, coat color of mice. So if you know about the introduction to monohybrid and dihybrid cross perfectly, then only we can solve these problems or the interaction of genes questions. Next subunit, that is multiple alleles, polygenic inheritance and pleiotropism. So here we'll be studying about different types of blood groups, Rh factors, and the type of disease, that is erythroblastosis fetalis, blood typing, blood transfusion, and what are the types of legal aspects of blood group genetics, and why, okay, we'll come to know why different blood group people cannot use the different blood group for the same person. Then inheritance of coat color in rabbits, isoalleles, pseudoalleles, and the position effect. So in allele, what gene or in which place the gene is expressing. So gene expression can be studied in this unit. Along with that, we will be studying about multiple factors or polygenes, polygenic interaction in man, that is skin color and eye color, why humans we are having different skin coloration. Some people are fair, some people are dark, some people are having medium. So that type of coloration, why the humans are having, that is mainly done in polygenic interaction. Then cytoplasmic interaction or the inheritance, where why shell coiling takes place in snails, and the kappa particles in paramecium, why the curve of the paramecium is in such a shoe manner. That all are studies done in polygenic inheritance. So these are the subunits which we'll be covering in the unit 1. So whereas in second unit, that is also genetics, where we'll be studying about in the first subunit nature and nurture. So why the genetics is important in nature, what is the purpose of studying genetics in along with the application to the nature, phenotype, penetrance, expressivity, along with the examples of experiments on Himalayan albino rabbit and human twins. Why the humans, like if you studied, if the same person or the person is having the twins of the same identical, we call them as monoidentical. Then identical twins. Then there are non-identical twins which do not resemble each other. So that type of studies will be done in first subunit. Whereas in second subunit we'll be studying about linkage and gene mapping. So here this is somewhat related to biotechnology and along with the genetics. So linkage, the complete linkage and partial linkage in Drosophila. What are the significance of linkage? Why linkage is important in humans? And there are some theories given by the different eminent scientists at what percentage linkage happens, what is the significance. Then linkage maps, how to construct the linkage map and chromosome map. Then there is a 2-point test cross, double crossovers, 3-point test cross. These are the studies that we'll be studying in the second subunit. Whereas in third subunit, we'll be studying about sex determination and sex-linked inheritance, types of chromosomes, that is sex chromosomes, then chromosomal mechanism of sex determination. So there are different types like XXXY, XXXO, ZZZW, ZZZO. So these are the different types of examples that comes over chromosomal mechanism of sex determination. Mainly we can study in humans, different types of animals along with the sex determination. Then sex linked inheritance in Drosophila. Here the Drosophila is having different characteristics for single organ. Like for example, Drosophila has white eye, red eye, yellow eye. So why the structure of the eye differs in genetic variation? So that we'll be studying here. And along with that, hemophilia and color blindness in man. What are the advantages and disadvantages of these diseases across the genetics? Then sex linkage in birds, sex determination of sex limited characters, sex influence traits. Why the humans? Usually some humans are partially bald, some person are completely bald. In humans, that we will be studying. Whereas in last subunit, gene and gene mutation and DNA repair, very important in studying the biotechnology and genes or genetics. Here we'll be studying concept of genes, definition, fine structure of genes like cistron, muton, recon, introns, regulation of gene expression, that is operon concept, very important, lack operon. Then mutation, there are different types of mutation like point mutation, frame shift mutation, inversions, insertions, and molecular basis of mutation. So what type of enzymes can be used for mutating the DNA or to isolate the DNA. Then there are some CIB techniques. So that will be controlled or completed in unit 2. Coming to unit 3, here it consists of human genetics and biostatistics. So if you are studying about the genetics, bioinformatics is a very important topic where you need to know what type of applications like mega softwares are there that will be seen. So in the first subunit of human genetics, here will be knowing about human karyotype, ideograms, pedigree analysis or making the family tree, then common human chromosomal syndromes, that is Klinefelter, Turner's, Down syndromes, which mainly affect the sex linkage in humans. Then there are some inborn errors like albinism, phenylketonuria, and sickle cell anemia, thalassemia, Huntington chorea, and there are some parental diagnosis, that is amniocentesis, then villus sampling or the chronic villus sampling, then genetic counseling. Why? For example, there are some therapists which they undergo or which they take genetic counseling of different age group person. Why they act? Maybe your father is very bright, why my student or why my son is dull? So that is genetic counseling. Then human genome project, that is also very important. So this is the complete analyzing unit or the subunit which comes in unit 3, that is first subunit. Whereas in second subunit, that is biostatistics, it is a very important, and here you will be studying about the introduction like to introduction to the biostatistics, presentation of data, how to compile the data and why it is important in the life science, and analyzing of data like mean, median, mode, and graphical representation, diagrammatic representation, pie charts, bar graph, or mainly making a linear graph, that we will be studying here. And along with that, standard error of mean. So while making the data in the field, you will be not getting complete 100% data, so there will be some error, so that is standard error of mean. Then there are some tests for significance, student t-test and chi-square test. So these are the important topics which will be covered in unit 3. Whereas coming to last unit, that is evolution and paleontology, here you will be studying about the theories which were given by Lamarck and Darwin. Darwin Wallace theory of natural selection, Lamarckism, synthesis theory or synthetic theory of evolution, Neo-Darwinism, Hardy-Weinberg law of equilibrium, factors that influence change in gene frequency like maybe gene mutation, gene flow, genetic drift, and in natural selection what are the types, then directional selection and disruptive selections, artificial selections also, isolation and isolating mechanism, like why the tiger they are mainly present in Indian subcontinent, why not in Russia and all. So you can tell there are some genes which affect along with the habitats also, that is why the organisms or the animals they are present in particular area at particular time. And there are some geographical isolations, reproductive isolations, ecological isolations, and there are mechanisms which helps in cytological, zygotic mortality, hybrid invertibility, and speciation, very very important, why a new species or when a new species is introduced to one particular land, why it is affected, so and why the population is getting decreased, so that is speciation. There are types of speciation, that is sympatric and allopatric speciations. And coming to last subunit, that is evidences. So here you will be studying about comparative morphology and anatomy of different strategies of humans, fishes, amphibians, birds. So when you compare the embryo of humans to invertebrates or the vertebrates like frog, the first embryonic stages will look alike, but when comparing them to last stages, the formation of fingers, the formation of beak, formation of mouth or the head, that will differ from one organism to another organism. So that will be studying in this comparative physiology and biochemistry. And comparative embryology and paleontology. And in paleontology you will be studying about the fossils, not much, but you need to study about the organism, that is Archaeopteryx, Peripatus. And in the last subunit, that is evolution of horse and man, you need to know how the horses started to evolve. When they were very short, then slowly started to grab their weight and height, and now they have become very much larger. First horses had fingers, now they are having hoof. Then evolution of man, you might have already studied little in your lower classes, that is Cro-Magnon and dwelling man and Neanderthal man, Java man. Along with that you will be studying about the evolutionary trends in horses and man. So this is the complete syllabus for the paper evolution and genetics. Thank you.